Welcome to the Goldman Lab Gene Expression Database. This is a quick video tutorial on how to interact with the platform. Please feel free to create an account, which will allow you to save and share your work. The registration only takes a few minutes. For now, we can proceed as a guest user. We can access the data through either the data highlights or by going directly to interactive visual analysis. Here, the first tab is Gene View. Our database is built largely around differential gene expression results from next-generation sequencing experiments in our lab. Here we see the data stored on the platform. The data are organized into three object types: differential expressions, gene sets, and Venn diagrams. Differential expression objects are derived directly from differential expression comparisons between case and control samples, depending on the experiment. In the name of the object, we see the case group first versus the control group, as well as the differential expression analysis software that was used there. We can also see the log2 fold change and the false discovery rate thresholds, and the number of up and down regulated genes, all with respect to the case group. Gene sets can be derived in a number of ways. They can be saved from differential expression objects, custom defined by the user, or uploaded from external sources. And finally, Venn diagrams are derived from the Venn View tab here, which we will explore later. So, to begin interacting with these data, we can type a few genes of interest into the Genes box and select the appropriate gene ID from the list. I can click through these genes here and see the relevant information on them. I can also copy-paste into this text field from external applications. You may have noticed that once we enter genes into the Genes box, some of the objects from this list disappear. It is because these objects are automatically filtered to only those that contain all of the gene IDs entered into the Genes box. For example, if a certain gene was not found as differentially expressed in a given comparison, that differential expression object will be filtered out. We can also go to the eye icon for any of these objects here. This presents additional information, and we can see all the case samples and the control samples that were taken into that comparison. We can also see the entire list of differentially expressed genes. We can pick a few genes from this list for further analysis. If we highlight them here and click Select, they are added into the Genes box. The list of objects here is further filtered down. You also may have noticed that I picked SOX10 twice, but the Genes box automatically removes duplicates. To see the expression data for these genes, we need to select a differential expression object from the list. For example, this one. Once we do have that selected, we can view the gene expressions plot that shows the absolute expression values and how they compare between case and control samples on both group and individual sample level. All absolute expression data on the platform are shown as log2 transform transcripts per million values, or TPM. Below, we can see the cell type expressions plot. Here we see the values for the selected gene from our current case and control groups and how they compare to other relevant groups in the database. This portion of the diagram is pre-computed by aggregating expression data from relevant and comparable datasets from our experiments. For example, the HD-OPC group represents all cell lines of this particular cell type that are derived from Huntington's disease patients, while the OPC group represents all unaffected controls. If I scroll back to the Genes box here and select a different gene, the two plots are updated automatically. Let's move on to differential expressions. Note that the genes that we have entered into the Genes box propagate into the New tab. Here, the work is concentrated on differential expression objects, And the list is not filtered. So even if our entered genes are not significant in a given differential expression comparison, it will still appear on the list. To create our first visualization, we can select a given differential expressions object. For example, the one that we've been working with before. On the volcano plot, we can see the relationship between transformed false discovery rate and log-to-fold change for all genes in the selected differential expression object. The genes that we have entered in the genes box are highlighted on the plot. The volcano plot is interactive. For example, if we wrangle up a few genes here, we can then select them from the pop-up window, and they will be added into the genes box. Note that I can select any number of genes from the volcano plot. I can either select a few individual ones from the list, or just all of them that I've wrangled up. Note that highlighting genes on the genes box also highlights them on the volcano plot. Next, if I select the second differential expressions object from the list, for example this one, this has the same set of case and control groups, but a different differential expression analysis method. The volcano plot disappears, but what I have now is a differential expression comparison table showing color-coded fold changes from the two differential expression comparisons for all genes that I have selected. And also differential expression comparison plot, showing the relationship between log-to-fold changes from the two comparisons. So here, based on this plot, I can tell that there is pretty good agreement between the two differential expression analysis methods. But if I select a different object with a different set of case and control groups, such a plot would, of course, would look a lot different. Finally, if I select third or fourth differential expression object, I only have the differential expression comparison table. So before we move on to the next view, let's quickly sign in. I'm now signed in with a test account, and I can go back to the interactive visual analysis. All of the genes that I've entered into the genes box are still here, and all my generated visualizations are still active. But now I can go back to the genes box, and I can save my entered genes as a new gene set. For example, gene set 1. Note that anything we save from within our account is only available from within our account and not from the public domain. So now let's go to the Venn view. In this tab, we can create a Venn diagram to see, for example, which of the genes that we were working with so far are also found in any of the existing differential expression or gene set objects on the platform. We can create a new Venn diagram from here and select any of the 4 available gene sets. For example, here's our new gene set 1 that we just saved, and we can select a gene set that was derived from a different set of experiments and click Create. Here we see that there's in fact 29 genes that are found in the overlap between the two gene sets. And these 29 genes are now entered into the genes box. Note that we can select and deselect any of the portions of the Venn diagram. And this automatically updates the genes in the genes box and the gene ontology enrichment results. So let's have these 29 overlap genes selected and go to the heat map. From here, we can select any of the available projects, and the heat map is generated automatically. We can order genes or samples either alphabetically or by hierarchical clustering. We can look at the color key for the values in the heat map. Finally, please feel free to share your work by either creating a bookmark and sending it to your colleagues, or by downloading an external report that will contain all of the generated visualizations and all of the entered genes from the gene books. Thank you for watching.